pops up in your mind, ask me, right? Anything that if I'm trying to get a sensing of how we are learning together, share with me your answer, right? Sometimes my questions are just simple yes, no's. You can shake your head, nod your head, or you can just whisper the answer from your mouth. I would love to see that response from all of you. All right, so please work on this with me. And to help you with that, right, let's not be strangers. A little bit about myself. All right, you can call me Teacher Jason or Mr. Palm, whichever is more comfortable with you, or just a very simple will do, right? So I've been teaching for a couple of years already. I hope I do not look like I have that many years teaching already. And it all started with studying bio in NUS. The university just simply called it life sciences for a fancier subject title. And I just thought it was cool to learn about how the brain works. So I studied a little bit about psychology as well. So it was a good way for me to understand how the brain learns things, how we remember things, how we make those connections in our brains. And then that just makes that learning process a little bit more enjoyable as well. And just like you and I have my presence in the classroom. I also have a life outside of the classroom. I exist outside these four walls. What I like to do outside there, just a little bit about myself here. I generally like being outdoors, and the pictures on the left there from my most recent trip to Korea in November. It was a beautiful autumn entering into winter. I caught my snowfall. It's pretty cool over there. And I do a little bit of rock climbing as well, and I have the wonderful opportunity of combining my passions for teaching and climbing. So you can look at the bottom right there. I'm running a climbing course at the Rock Wall at NUS, right? And most recently, I still do play in a band. So I still enjoy making music for. So some of you may also be in a band and enjoy making music as well. I just had my concert on Sunday, and I'm still recovering from that post-concert high, as you can see in the picture in the middle there as well. Right, so whether I see you out hiking in the woods, climbing in the gym, or just enjoying a music concert there, if you do see me outside, please do say hi. But if not, right, so just know that we are here to learn together, right? Just a bit about myself. There's so many of you right now. I will take my time to get to know you as the weeks progress and when we learn together next year. But for today, this is me. And what about the center then? A very brief overview, right? I am at my classes are at these two places, Kovan at Tampines, sorry, only. And for all the other centers, right, our newest one is Novena. Why is that of note? Because our current Sec Force, we are going to be doing a practical session with them, like the real Bunsen burners and titrations and stuff for them at our Novena outlet. So that's what we are working on. And when I do see y'all in my classes next year, that's something we can look forward to as well. Not necessarily has to be in Novena, it's just something that's available for us because hey, this classroom is big enough as well. If we get a good enough number, we are going to be able to conduct it here as well. Right, so there's something for us to look forward to here. We're going to be working on content, we're going to be working on analysis, we're going to be working on how to remember, not to remember, right? The how is more important, I feel, right? Because I can help you with the how, I cannot help you to remember. That's on you, isn't it? Right? Okay, so that's what we're going to be working on together. This is just saying that we are good. All right, but that's for you to decide, right? Whatever I say is for you to decide still. Okay. I mentioned that already, all the practicals and assessments and stuff like that. So therefore, let's jump right into it. Okay, so I hope they all have the materials with you already. So we're gonna start on to looking at the contents page over here. Now, at this point, you're looking at these three topics. There may be a scenario where some of you may not require one or two of the other topics for your end of years. If that is the case, allow me to help you understand this perspective like, hey, we're working towards the O's next year anyway. Okay? Right? So whatever little tidbits I can share with you, how can I better understand this concept? How can I better keep this in my head so that when I do learn this next year, hey, I have a better step up already. Okay? Right? At the very most, I think only excretion could be something that we may not require end of years. So at this point, who doesn't require excretion for your end of years? Oh, okay, not too bad then, right? So all of us need transport and respiration in humans. Yes? Yes? Thank you for your nods. Thank you. All right, so allow me to share with you what I can give you better insights on these three topics together. Let's jump right in. Transport in humans. Red blood cells. Nothing too fancy. Of the three unique structures and features. Of the three unique structures and features, can I just ask you all to look at those 3 points there? Which one do you think is the most important feature for the functioning of a red blood cell? Containing the hemoglobin, isn't it? Thank you. It is to contain the hemoglobin because, let me challenge you with this idea. If the red blood cell doesn't have hemoglobin, no matter how biconcave, no matter how empty it is inside, it's not going to be able to function, isn't it? Excellent. Thank you for that, right? So it contains hemoglobin. So how can I help you understand this better? We are looking at the word that comes up next, reversibly. We need to mention a reversible binding in the function, right? So understand reversible, right? Bear with me, I'm just trying to remember the names. The first thing I see over here is Anusia. Anusia. What's reversible binding? Do not use the word reversible to explain reversible. Fine, I'll take that. We can release it. And that's important, right? Imagine all of you are red blood cells, raring to function, deliver that oxygen there. When you get to the lungs, you need to be able to bind to. But when you get to the muscle cells, you need to be able to let it go. Right? So you need to be able to have that reversible binding. And this actually reminds us, hey, when it comes to respiration in humans, there's something that we should think of that binds to hemoglobin as well. There's something that binds to hemoglobin as well. Sonny, help me out. What else binds to hemoglobin? Wonderful, thank you. So that falls under the harmful effects of cigarette smoke, isn't it? Carbon monoxide will bind to hemoglobin. But what's the difference here now? What's the difference here now? Yuxuan? It is not reversible. In other words, it is going to be used either of these two words, irreversible or a permanent binding. And therefore, hemoglobin is so madly in love with carbon monoxide that it will not let it go. And with that happening, you have lost that one hemoglobin's ability to transport oxygen. Okay, so we will come back to this idea. We talk about respiration in humans later on. Thank you, Sonny. All right, so coming back over here, fairly straightforward. We are looking at biconcave surface area to volume ratio. Surface area to volume ratio. Here, diffusion. So this is one of those things that even though your topic list does not include movement of substances, I would very much like for all of us to know that this topic cannot run away one. It appears in so many topics, even in a handful of topics to come next year. So diffusion, osmosis, what's the third one? Wonderful, thank you for that chorus. Active transport, these three things, right, cannot escape one. So it will do you good to make sure you have a good foundation in this topic. Okay, wonderful. Let's move on. White blood cells, white blood cells. Okay, so just now for a blood cell, primary function is to transport oxygen. Primary function of the white blood cell is to protect. It's to protect, simple as that. Let's just keep it very, very simple, protect. How do we do so, right? So here in our syllabus, we are only focusing on 2 different white blood cells. So actually at this point, we rarely would be writing down the words white blood cell in our answers really. When a question asks you, hey, this cell in the diagram, what is it? We rarely should be identifying it as a white blood cell. We should be choosing from either of these 2 words, the phagocyte and the lymphocyte. Okay, the functions, I've given you the words there already. But if you're given a diagram, how do I identify a phagocyte and a lymphocyte from the diagram? How are they different in their appearance? How are they different in their appearance? Chloe, ow. Any insights to share with us, please? What's the difference in their looks? What's the... Okay, some stuff poking out, Trisha, you know what your friend talking about or not? I kind of know what she's talking about because I know what I'm looking for, right? Marzuka. Marzuka. Who's Marzuka? Oh, I got another friend. Oh. What was she talking about then? Hey, Chloe, your friend don't know what you're talking about. Okay. Zihan. Did I say it right this time? Zihan. Hi. How do we identify a phagocyte? The nucleus. But first, do you know what she's talking about? Okay, tell me about the nucleus then. The phagocytes. How do we describe the nucleus? Excellent. Thank you. Come, let's write this down here. Looped nucleus. Chloe, you're not wrong. I'll get to that. Give me a second. So phagocytes have a looped nucleus. So if I were to draw it very briefly, okay, I'm not gonna score any points for art. But what's the shape of the nucleus of the phagocyte? We have three kind of like segments, but they're all connected. So there's one nucleus still. Can we see that? Right? So it is still connected like that. Okay, later I show you a diagram, okay? Don't frown at my drawing like that, lah. I'm doing my best, okay? So you will see a diagram later on. But that black stuff over here, instead of, we see this as a nucleus in a very standard cell, isn't it? Right? But now the nucleus, can you just think of it as you're like squashing it and spreading it out like that? You all used blue tack before? You all squash and pull, right? That's it. Okay? Right? So that's the looped nucleus. So instead of giving you this for phagocyte, that for lymphocyte, can I just make it easier for you? You will see this structure, phagocyte. If it's not, then lymphocyte lah. Okay? Much easier, isn't it? Thank you. Right? But if you do want to see a diagram for it then, the lymphocyte, right? What do we say about the nucleus here then? 30. Excellent. It has a humongous nucleus. Compared to what we generally see, it's like, okay, it is a large organelle, but it's not going to take up as much space as now, isn't it? Right? So we use that to help us remember that. The words looped nucleus are words that are used to describe the nucleus of a phagocyte, but the word humongous nucleus is not what we write down, okay? It's just for you to remember. Lymphocyte humongous nucleus. Okay? So let's come back to Chloe's points, right? Like, what's this? Okay, so this actually helps us to understand the function of a phagocyte. Now, we have written the words down over here, phagocytosis. Actually, can you describe phagocytosis for us, please?
Where it like makes the foreign matter not harmful for the body.
When it makes foreign matter not harmful for the body is literally what all white blood cells do. So, Chloe said, the phagocyte got like that thing, right? I want you to look at these words here. To ingest or to engulf. If you focus on the word engulf, I want you to think of the phagocyte giving the foreign particle a hug of death. What is it going to do? It's going to pinch out its cytoplasm and then engulf the foreign particle like that. So what Chloe was talking about is this thing. The cytoplasm can stretch out. It can pinch outwards. And then this part here, connect back. So now I've encircled you, I've engulfed you. I've given you a hug of death. But in this case, all I'm doing is to isolate the foreign particle. That's not quite enough, isn't it? After you isolate already, you're still back. So what do I have to do next? Phagocytosis is a two-step process. After you engulf the foreign particle, I want to also thank you for not saying killing. I heard some killing there, right? But we're going to break it down as well. So phagocytosis, number one, you have it over here. You choose which word makes more sense to you, to ingest or to engulf. Your number two, you either go for digest or you go for breakdown. Natalie asks, the phagocyte, the three circles connected, it refers to the nucleus, but take note, it is just a very, very generic drawing. Sometimes you will see two, sometimes you will see a little bit more. Right? So it's not a fixed thing. It's just a general diagram. Clear? Natalie, have I answered your question? Awesome, thanks for asking. Okay, so, all right. Some of you may have learned chapter 9. What is the group name for these bad things? Pathogens. Excellent. Pathogens. Okay, pathogens, right? We learned that in topic of infectious diseases in humans, isn't it? Okay, infectious diseases in humans. So therefore, phagocytes are the ones that actually go down to the battlefield. Hug of death. Goodbye. What about the lymphocytes then? Now remember we say they have a large nucleus, isn't it? So this is where the big brain moment comes in. Instead of rushing down to the battlefield to go and fight, they stand far away and shoot arrows only. So they are well protected from the fight. Big brain moment, right? So they have humongous nucleus. This allows us to understand as well, hey, what are these arrows that they are shooting? They are going to be releasing antibodies, right? They're releasing antibodies. Now, a very generic description of the function of antibodies is simply bind to and neutralize. Done. Okay? But this one is not bind to and neutralize the pathogens. It is the toxins that the pathogens release. So there are two ways for us to go. I could be focusing on the toxins that the pathogens release, or I could be looking at the pathogens themselves. Right? So that's something I want to write down together with all of you then, right? So if here is number 1, what color can I use? Bind to neutralize toxins is number 1. What would number 2 be then? What else do the antibodies do that helps with this protection of the organism? Excellent. I want that word, clumps. It clumps the pathogens together. Clumps pathogens up. But for what purpose? I want you to imagine when you eat rice, that one bowl of rice, do you eat one grain at a time? No, right? But you use the spoon to grab a whole chunk. So that is the clumping one. Much easier to eat, isn't it? So now you are the phagocyte lah. If you eat one grain at a time, wow, tomorrow you haven't finished your dinner. But if you eat a whole chunk at a time, much faster. And that's the whole idea as well. The lymphocytes producing the antibodies. Step number 2, clumps pathogens up. Helps the phagocytes function more effectively. Okay? So it's linked, right? Clumping up together. All right, white blood cells done. Okay? All right, at any point, questions, please let me know. Gonna move on to our platelets. All right, gonna move on to our platelets now. So, platelets. So, for red blood cell, primary function, transports oxygen. White blood cell protects the organism. For platelets, we are going to be thinking of blood clotting, right? But take note, the word that I did not highlight is actually pretty important as well. Platelets initiate. They get it started. They do not do the clotting. A very big difference, isn't it? They do not do the clotting. They get it started, right? Once they press the button start, their job is done. Okay, so very, very important there. For clot formation, please remember we are always going to be thinking of in and out. We're always going to be thinking of in and out. The clot forms to prevent good stuff from exiting. The clot forms to prevent bad stuff from coming in. Use that. Everybody can remember in and out, isn't it? So help yourself with that. All right, help yourself make it easier like this. So how does it do so then? Now, over here, I've listed a very generic one, but for pure bio, we need to expand upon this. Pure bio, we do need to expand upon this. So, Natalie, same time. Annabelle, hi. What's that enzyme here? You pronounce that word correctly, but that's not the answer. It's the one before. The one before. Kaylin? Hi. What's the enzyme? Starts with a T. I'm seeing some mouthing, but that's not the correct answer still. Thrombin is not what we're looking for. Thrombokinase. Thank you. Thrombokinase. Wow, I never hear before. Who never hear before? Who never? Okay, come. So therefore, we have words over here. Let's use this space at the bottom. Let's draw a waterfall together. Okay, so here we have at the wound. Okay, by waterfall, I mean the diagram one is a waterfall of words. Okay, let's write it down together. So what happens at the wound? Please start the journey with me. Platelets. What do they do? Platelets and damaged tissues. What do they do? They release thrombokinase. Now, if I did not tell you what it was, I just give you this word here, will we be able to figure out what kind of a biological molecule it is? We can. Just from the name here, can we figure out what kind of a biological molecule this is? The ASE tells us that this is an enzyme, and all enzymes are proteins. Thank you. Okay, link. The more information we link together, the less separate things we remember. Okay, awesome. Thank you. So what does thrombokinase do now? Then we bring in the words that you all were whispering to me just now. Aiden, what do you say just now? Thrombin, but thrombin doesn't start as thrombin. It actually gets activated into thrombin. So thrombokinase's function is to activate this into thrombin. It's very simple. Prothrombin. Okay, prothrombin. 1%, not hate 1% shake head, since you see before. Okay, let me finish this cascade first. Well done, Joel. Thank you. You got them both. Let me finish this cascade first, then I will share with you a little bit of like why some of you have heard of it and some of you never heard of it before. Let me add something cool here as well, because calcium ion. So this activation of prothrombin into thrombin requires calcium ions as well. Okay? Come, work with me first. Later on, then I will share with you why some things I'm sharing with you like this. From thrombin, easy already. Everything from here on is something that I'm hopefully everybody can remember. What does thrombin do? Very good. Soluble fibrinogen into insoluble fibrin threads. This orange thing everybody know, right? Yes. Thank you for the nods, right? Insoluble fibrin threads. From here, insoluble fibrin threads. We're one step closer to the formation of that clot already. What is a clot? It's a mesh of what? So the threads now, the reason why I want to write down the word threads and the word or the word strands is for us to know those wriggly lines, they will come together to form a mesh. What does the mesh do? Trap, right? So the idea of a fishing net, a fishing mesh, is going to trap stuff. We're not fishing for fish, we are trapping red blood cells. That is the clot. Okay? All right, so we are going to form a mesh. Now we have a clot. Now we have a clot. And this is what I meant by the waterfall. This is actually referred to as a clotting cascade. For those who are looking forward to continuing to learn this beautiful science in JC next year, you'll hear the idea of cascades more as well. So the cascade happens because from here, the moment we initiate it, the moment we start it, it flows like that. That's actually also something I've been trying to get us to understand as well in our learning. When we remember things, remember everything at the start. Everything flows. Right? Okay. So from some of your notes, I'm glad that you can understand this rationale of remembering things. The more we can link them together, the easier it is for our memory to flow. Okay, so over here, then we have a clot. So a couple of things some of us may not have been familiar with would be thrombokinase. So this part that I'm circling, right? So this part is something that we may not be familiar with. At the very least, right, thrombokinase, I think is fairly straightforward, right? Because it's just going to be an enzyme, is what they release here. Now, these are things that maybe could appear as an application kind of thing. We have seen this appear in our questions before as application. So I leave it up to you then, right? We can kind of see it flowing like that. So it's a simple enough thing to add into what we have learned before. But if you really do not want this to add on to your mental load there, that's fine as well. Okay? So as long as I shared this with you, you kind of have it in the back of your mind. So if we do spot it so accurately, it does appear in the end of years. Hey, you kind of know a little bit more about this already. Can? Right? On that note, I assure you it will definitely help for our O's then. Was that a question? Does the mesh also trap white blood cells? Being a mesh, it like logically it's just going to be like a barrier, isn't it? But there's something very unique about white blood cells. There's something very unique about white blood cells. They will not remain trapped. Why? What feature am I looking for in the white blood cell? What feature am I looking at then? Sonny? The shape. Very good. And if you can see, they can squeeze through the gaps, then they can wriggle their way out of the mesh also, ma. Can? Right? So some may get trapped, but that's not the main purpose. Right? The majority component of the clot are the red blood cells. Okay? So before we move on from clot formation, what is a scab?
Right.
Okay, excellent. I love that. Right. We want to know what is the meaning of a scab. So Alexis is telling us that it is a dried blood clot. So then, Alexis Tan, right? What must happen to the clot for it to form a scab? So there are two things we need to know. She already told us dry out means must lose moisture. There's one more thing. Oxygen. Wonderful, Ashley. Thank you. Okay, so clots. If they lose water, meaning allowed to dry out, and then react with oxygen in the air, they will form a scab. So at this point, looking at the stuff in yellow, scabs only form if the wound is external, is superficial. But clots can form inside and outside. Clear? Please take note, if it's an inside wound, so your blood vessels inside, not the one on the skin surface. If it's an inside one, got cut there, can a clot form? Absolutely. But can a scab form? No. Only if it's the outside one, then we have that. Okay? Alright, so be very clear about it. Be very clear about that. Wonderful. That's it. Let's go. So plasma. Now plasma. This is the fourth component of blood. It is the only acellular component. It is not a cell at all. And I typically help us to remember plasma as soup. It's just soup with all your dissolved things within. Right? Just that in the soup that we drink is all good stuff only. But here, got some bad stuff lah. Can? Right? So on this note, let's look at the bad stuff first. Urea. Urea. How is urea formed? Fiona. Who's Fiona? Fiona. Hi, Fiona. I'm looking for a D word. I want to start my description with a D word here. Charlie. Thank you. Deamination. Deamination. Lauren of. If you say deamination of amino acids, no marks. You're missing a word. Yes, excess amino acids. Thank you. I'm not done, but let me just pause here. The idea of deamination is to break it down, right? So why is the word excess so important? You're not gonna break it down if you still need it, isn't it? So the word excess is important. Clear? You're not gonna throw away food if you're not done with it yet. Okay? So excess amino acids is very important. All right. Deamination of excess amino acids. Nickel in the I always love giving this clue. Look, very good. Liver. My drawing of the liver is so accurate, isn't it? In the liver. Liver, if you did not know, is a triangular shaped organ. Where? Do you all know where your livers are? Where is the liver? Where's your liver? Can you just roughly point to your livers? Roughly point to your livers. Do you know where your livers are? That's the amazing thing about bio. I learn about yourself. Where are your livers? Okay, liver. Where your liver? You say where's your liver? You like that? That's just very generic leh. Which side? Left or right? Left or right? The liver is on the right side of your body. Okay? So, hey, this is my right side. Mirror, opposite. My right side, you find your lowest rib, there is your liver already. Okay? Your liver is kind of there. Right? Find your lowest rib there. Why, Annabelle? Is it lower or higher than expected? Is it there? Right? Also, liver is somewhere there. Okay? Your liver is on the right side of your body. Right. Deamination of excess amino acids in the liver. So what happens to urea? Oh, I think I heard the word already. Who said it? Was it you? Excreted. Excreted. Maneuver. Hi. In. No, urea is formed via the deamination of excess amino acids in the liver, and urea is excreted in urine. Yes. Okay, urine. All right, urine. Okay, so that's for urea. We'll look at it again deeper. We'll talk about excretion later on as well. Okay? Alright, for now, that's it. Now, for all the useful things in the plasma soup that I mentioned, there is no need to just look at this whole chunk here and have to memorize spelling like that. Do not need to do that. As we progress in the future topics, you will become more familiar with these things here. For example, you recognize this? What's estrogen? A hormone. What does it function as? For which system in the body? Reproductive. Thank you. Right, estrogen is a female sex hormone that we will learn in greater detail when we come to the topic of sexual reproduction in humans. Okay? Right? Testosterone is the male sex hormone then, right? So again, that's that. But for insulin, this one we know already. So therefore, insulin. Two effects. What are the two effects of insulin? Nico? Two effects of insulin. When insulin goes to the cell, what is the message that insulin sends to the cell? That is the overall function, the overall purpose of insulin is to result in a blood glucose concentration going back down. How? Zihan.
Um, so
How's that? Why suddenly tell me pancreas? I don't need pancreas. Insulin? How's that? Insulin. Okay, Zhan, tell me first, what are the target cells of insulin? What is target cell? So if I am insulin, I'm going to traveling throughout the bloodstream, isn't it? Who do I go to? You got brain here, got stomach here, got lung there, got da 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 da. Who do I go to? Liver and thank you, thank you. Okay, all right, but liver and muscle cells. So the target cells of insulin is liver and muscle cells. First and foremost, please remember that. We have liver and muscle cells. Okay, liver and muscle cells. But hey, my first question is still unanswered. The two effects of insulin. Actually, you did one first.
makes the cells more
Two
absorb more glucose.
to glucose. Thank you. You cannot just stop at more permeable. It has to be more permeable to glucose. More permeable, that's why I can absorb more. Right? So let's combine that into one. Right? So insulin tells the liver and muscle cells to be more permeable and therefore absorb more glucose. Okay, function one, effect one. Noah says to increase metabolic rate. So that is to tell the cells to use more glucose. Yes, we have that as well. Insulin has, if you're looking at it whole, it's like 4 or even 5 effects by itself. What I'm looking for are the two more prominent ones that will result in a more significant, a more drastic change in blood glucose levels. So we are focusing on glucose. To glycogen, thank you. Right? Look at that, right? If the cells absorb more glucose already, then the cells inside got a lot of glucose, right? What is affected now? What is the topic that I told you all will always appear on? Concentration gradient, isn't it? If the cell keeps on absorbing, what happens by gradient now? Becomes gentler until I reach what do I call this point where both sides have the same equilibrium? Thank you, people. So if it's at equilibrium, can I continue absorbing? No. Do I want that to happen? Absolutely not. So therefore, all the glucose that I have absorbed, I'm going to convert into glycogen. So this also helps me to maintain my gradients, isn't it? Okay? All right, let's write that down, okay? So increase conversion of glucose to glycogen, okay? Right, increase conversion of glucose to glycogen. Yes, Chloe, right? It does stimulate the liver to do this, okay? So gotta keep that in mind. These things here, always remember, we are also linking topics. This is in biological molecules, this is in digestive system, isn't it? The more we can link together, the less individual parts we have to remember. Okay, let's move on. So we're done with the 3 blood cells, we're done with the plasma soup. Let's have a look at our blood typing, right? Blood typing appears in this topic here, as well as another topic in future that we have not learned yet, but maybe you have heard of it. Any guesses as to what that second topic is that blood type will come out again? Reproduction is involved, but it's not learned in the topic of reproduction. Genetics, molecular genetics involved, or not under the topic of molecular genetics. It is a very closely related one though. To pass down. Inheritance, right? So in the topic of inheritance, we'll be learning about how a parent with this blood type and a parent with that blood type can result in offspring with this, or that blood types. But that is a story for next time when I do see you in class next year. But for now, the human ABO system. right? ABO is just the name. There's an arbitrary letter we assign. Over here, very simple. Look at the first column with me, the first two. So antigens, can I ask you to remember this as agents on the red blood cell then? All right. The red blood cell contains these tiny little antigens there. So they are on the red blood cell. Well, the other word here is in. There's a difference over here. Antigens are on the red blood cell, and that's actually what we use to assign your blood type. If the antigen on your red blood cell is assigned as antigen A, your blood type is blood type A then. If your antigen is B, your blood type is B. If you have both, you are AB. If you have none, you're O. Simple as that. Okay? Right? Assign yourself to that. Now, a very big clue that I'm going to give you right now is to think of enzymes. They are not complementary. Thank you. They are not complementary. Well done, right? So, and this is important. They should not be complementary, because what happens if this antigen and its complementary antibody meet? Agglutination. Wonderful. Thank you. What is agglutination? Thank you for saying the word clumping, right? A very commonly wrongly used word is clotting. And we just talked about clotting, isn't it? If you want to talk about clotting, that whole cascade must be involved. All the way from thrombokinase to the fibrin threads. Okay? Right? So must have that. So over here, well done, Chloe. Clumping. And we have it over here. Right, at the bottom of your page there, if we have antibody and antigens that are complementary, they will bind together. Then all the red blood cells will be like, yay, party! Then they will form a whole clump there. But it kind of functions similar to that clot, which is why some people will confuse them. Okay, so well done for not confusing that. So very important, we're looking at matching blood donors to recipients. First thing I helped us to recall is use antigen to determine blood type. And then whatever you have in the plasma is going to be the opposite, the non-complement, isn't it? Right, use that. Then when we talk about receiving and donation, that is something that we have in our questions as well. You could just go ahead and remember this, or simply stick to the guideline is you can always donate to and receive from your blood type. Plus one more thing. Plus one more thing only. That's all. I can always receive from my blood type and O. I can always receive from my own blood type and O. I can always give my blood type and AB. Okay? So therefore, the words that most of us would have been exposed to already, universal donor and recipient. Thank you. The universal donor is blood type O. Universal recipients are blood type AB. Okay? Right? Pretty straightforward there. The idea of agglutination could even come up when we look at the topic of sexual reproduction in humans, when we look at fetal development, right? When the fetus is developing in the mother's uterus, could agglutination happen? Keep that question in your head. We'll look at it next year. Let's talk about it. All right, done with blood typing. Blood vessel is done. Easy peasy, right? A for artery. A for away. Done. Okay? Right? You could look at these two blood vessels, arteries and veins. They are parallels because they both serve to transport. They both serve to transport. What word do I want to highlight for capillaries then? Exchange. Wonderful. Thank you. Okay, so we compare like to like, ma. So if artery and vein are the two transport vessels, then we compare them. We don't go and compare suddenly to the capillary, isn't it? Right? So look at the two transport vessels. Look at the transport vessels. A for artery, A for away. You could very simply, if A is artery, A for away, then vein is the opposite. Done. But if you really want to look at it, right, you can see veins bring blood in to the heart. Okay, that works as well. Can? Right? Either way, both bring us to the same conclusion. And then looking at capillaries, exchange. Does a certain topic come to mind again? Movement of substances, right? You have diffusion, your osmosis, active transport. Look at that. The moment we can keep on linking, good already. Why is the capillary so good for exchange? Why is it? What are the adaptations it has? What are the structural features it has that makes it adapted for its function? Right? So we talk about everything you learn in movement of substances. You have your concentration gradient, you have your surface area to volume ratio, you have your barrier thickness, da 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 da. So therefore, you look at the structures over here. We look at capillary, since we were talking about capillary. What is an endothelium? What is the endothelium? The wall. Thank you. So therefore, in your syllabus, you could just say wall. But the name of the wall is the endothelium. Okay? It's the name of the wall is endothelium. All right. But we are going to make a correction. Not correction, make an optimization. Instead of just saying very narrow. Your very narrow and my very narrow subjective, isn't it? So let's write down something objective. Look at my beautiful drawing once more. This is a capillary. This is the capillary cell. So this is what I'm drawing, isn't it? It is one cell thick wall. The wall only one cell thick, so very thin ma. I mean, what else can be thinner than one cell thick, right? Okay, in a structure like that. So that's why the wall is one cell thick only. So now, what is the header in the second row there? What's the word there? What's the L word? Lumen. What's a lumen? The space inside. Very good. So the lumen, if you look at this, it's a blood vessel. The space is the lumen. Okay? So how wide is the capillary lumen? We're not going to use subjective words. And we're going to write down something very objective. When we write this down, there are no confusions. Oh, really so small only. The clue I'm going to give you next here is, what am I drawing? Red blood cell. One cell. What's the word?
One.
Thank you. The lumen is one cell wide. The wall is one cell thick. And well done. Thank you. Therefore, instead of very narrow, we say one cell wide lumen. So therefore, capillary, just one cell, one cell. One cell what? The wall, one cell thick. The lumen, one cell wide. Can? So make sure you don't go and flip. Okay, couple of questions here. Ray, epithelial is for external surfaces. Your blood vessels are internal surfaces, so endothelial. Natalie, diffusion and osmosis does occur, right? Go and revise the definition for diffusion and osmosis. They are for different particles. Okay, so capillary done. For arteries and veins, let's hope to get through this very quickly. Why do arteries? Thick walls. It's something about the high blood pressure, but what about the blood pressure? Wonderful, thank you, three of you, right? So a very common comparison is artery versus veins. Why do arteries have thick muscular walls? The word we're looking for here is to withstand the high blood pressure. Let's flip it around then. Artery lumen very narrow leh. What for? Artery is the wall very thick, withstand the high blood pressure. The lumen very narrow. What for? Kind of there, but no marks, because the only way we can increase blood pressure is due to the action of the heart. Only the heart can cause blood pressure to increase. So it's about, yeah, kind of there. We do not increase blood pressure, but rather to, same word there, cannot. Maintain. Wonderful. Was it then? Thank you. It is to maintain. It is to maintain. Hey, I want blood to be able to shoot out across the entire distance in my body, isn't it? Right? I do not want it to drop in pressure, because the drop in pressure means a drop in speed as well. And then your cells all the way at your feet are going to be very sad. Not enough oxygen and glucose. Right? Thank you, boys. Right? So therefore, yes. Okay, so use this idea here, right? So thick wall, withstand, tahan, very strong, won't burst. Can? Right? Maintain, keep it nice and narrow. Well done, Noah. Okay. Then for vein usually the question asks us to explain and compare in terms of artery. Then we use this way. But if the question asks why do veins have thin walls, you don't start with maintain or withstand. You start with the low blood pressure first. That's why I don't need. Okay? Right? But with the low blood pressure comes another problem. The blood has no shepherd. So therefore, blood can backflow, right? How do we prevent that from happening? Okay. V for veins. V for valves are present in the vein. Now, this is a very, very simplistic diagram of a valve in the vein. Can I just ask you all to have a look at this first? A very simple diagram of a valve in a vein, isn't it? Right? So at this point, I'm just going to do this. Use my hands here. I'm going to form a heart shape. I'm going to open it up, right? So over here, my hands are going to form that valve over there, right? So the flaps of the valve are always going to be pointing in the correct direction of flow. Why? Imagine a unit of blood over here is going to be able to push the valve open. But if backflow were to occur, what happens? It's going to push the flaps towards each other, snapping it shut, preventing excessive backflow. Okay? So that's what happens in the presence of the valves. Clear? Right? That's exactly it. And this valve action is also found in the heart. It's the same thing. The same rules apply. Okay? So when we talk about the heart now, we have the 4 valves. Can we see that the valves, the flaps, are also directional? They are always pointing in the correct direction of flow. Right? So we have on the diagram over here, please make sure your left and right whenever we see a diagram of the heart in our questions, imagine that person's heart, that person looking at you then. So for example, this heart like this, it belongs to me. This is my right hand, then this is right side. Clear? Right? So the heart, please make sure to orientate yourself properly. Okay? So the tricuspid valve over here, flaps pointing this way, correct direction of blood flow from RA to RV. Now, whenever we come to this diagram, absolutely understandable. It's like, wow, so many things. Do I have to remember every single thing over here? All right, give me a sec first. Ray, do not need to use the word endothelium wall, works as well. Natalie asks why arteries no valves. Do you know why? Yeah, the blood pressure is so high. There's so much pushing force. There's no chance for you to walk backwards. Okay? Just imagine yourself trying to swim against a waterfall. Not going to be able to do that. Okay? All right. So in this diagram over here, super overwhelming. So therefore, let's use this space on the left-hand side. Let's going to draw the heart. Right? And when we look at this heart here, the chambers, we have 4 of them. And you realize that my horizontal line is not drawn halfway through because I want to show that. What letter is this? The V, isn't it? Use that. Because why? This are the ventricle. Okay? Right? And then next thing, very simple. Everybody learned about the alphabet. The alphabet starts with the letter A. Right? The letter starts with letter A. So, atrium, ventricle. And like I said already, please make sure get your left and rights correct. That's it. So I'm still sticking with the idea of the alphabet starts with letter A, right? Because blood is going to do this. The journey of the alphabet starts with the letter A. The journey of blood in the heart starts with the letter A. It's going to get out the other side. That's it. right? It's always single direction. I'm going to do it this way. Direction same, right? Direction same, Enter at atrium, exit at ventricle. What have I changed in my arrows here? My colors, isn't it? What am I trying to represent here? Thank you, right? Oxy and deoxy. Obviously, red is oxy blood, blue is the deoxy blood. So at this point here, I hope we can also remember. Have you used this before in school? What does it represent? Thank you. The left side of the heart, oxy blood, right side, deoxy blood. Okay? Left side of the heart contains oxygenated blood, right side of the heart, deoxygenated blood. Okay? Right? Use that. So this is where we combine naming of the vessels as well. Vessels always have a two-part name. First name, last name. My name is Jason Pang. So blood vessels got first name, last name as well. First name is always organ association. Second name is always direction to or from the heart. So the second part, right, is always the simpler one. Always tackle that first. A for artery, A for airway. Okay? Right? Then the first part is organ association. In our syllabus, we learn 4 organ association names. Can I ask you to just peruse it in your head first? Scribble it down on your notes there. What are the 4 words for organ associations? There are 4 different ones. Renal, pulmonary, hepatic. The last one is the one that people struggle with. What is that? Pulmonary has been written. Hepatic is still there, so it's still liver. Thank you. Well done. Coronary. Come, let's write it down. Pulmonary to associate with hepatic, renal, and last but not least, you say heart, then wrong already. Why? Then coronary artery, heart to heart. Yeah. Cardiac muscles, heart muscles. Thank you. Okay. Be very clear, be very clear. Because heart to heart means circle, and that doesn't happen. Okay. So for our syllabus, it's just going to be these 4. They are obviously not exhaustive, but this is what we need in our syllabus only. Fair enough. We can get to these 4. Great. Maybe just one thing I can point out since we are on this diagram over here. These 2 things I'm circling. Let me see. What's the difference here? Okay, so both of them are vena cavae. They are the same kind of blood vessel. But superior and inferior does not refer to one being better than the other. It is simply words that we use in anatomy for positions. The superior vena cava brings deoxy blood from above the heart into the RA. The inferior from the bottom. Right? It's simple, your heart is here, isn't it? We have organs above the heart, so therefore, via superior, via inferior. Okay? Very straightforward. Your syllabus just requires vena cava. Let's move on. Okay, so cardiac cycle graph. Graphs are something that for the most part, people see graph like, oh my goodness, I gotta do some math already. From the smiles I see in the class here, I can see that that's what we are working on. So allow me to help you understand this better, right? Instead of looking at the entire graph, my immediate suggestion is go to the intersections. The intersections are where the magic happens. The questions will usually ask you about intersections. Okay, so therefore, I'm just going to list down some intersections over here. I have I have A, B, C, D like this, right? If you can go ahead and label them like that with me, then easier for us to talk. Right? A, B, C, D, the intersections, just focus on the intersections. And in my graph, I have it nicely colored. Blue for LA, red for LV, black for iota. Okay? Lauren, choose one intersection, please. Choose one intersection. A. Okay, looking at A here. So focusing on A, is between blue and red. So that is for LA and LV. So the y-axis here is in terms of pressure, isn't it? At intersection A, whose pressure is becoming greater than the other? The LV's pressure is becoming greater than the LA, isn't it? So having listed down LA, LV, the next step to do is use your inequality symbol. Can we understand that? We wrap it up with P for pressure. And there you have it. Once we can arrive here, what is the name of this valve here? Identify the valve between LA and LV, please. What is the valve between LA and LV? The bicuspid. Thank you. And how you recognize whether the valve is open or closed? You simply look at the inequality sign as well, but this time, read it as an arrow. Blood flow from LV to LA, correct or wrong direction? LV to LA, correct or wrong direction? Wrong. So what must the valve do? Look at that. Once you've written all this down, let me eyes on the screen. Let me walk you through this sentence here. How do we make use of these 3 steps I've just shared with all of you here? The bicuspid valve closes and the pressure in the LA becomes smaller than the pressure in the LV. That's it. Apply this exact same 3 steps to all 4 intersections. You will be able to find when your valve opens, when your valve closes. You could choose to do it 4 times at all 4 intersections. I already made it so much simpler, isn't it? Identifying intersections. But let me make it even simpler for you. Instead of looking at 4 intersections, you only need to look at 2. If blue and red intersect here, the valve closes. Blue and red intersect again here, and the opposite, isn't it? Intersect here to close, they intersect here again, open, no? Fair? That's it. Right? So I hope I've brought everybody one step closer to being comfortable with this graph over here. Look for intersections, that's the first thing. Then what do you do with the intersections? Compare the pressures. Use the inequality as an arrow. That's how blood flows. Correct, open. Wrong, close. That's it. 10. Last thing before I move on from the valves over here. Why? Why will the pressure in LA become smaller? Why will the pressure in LV become greater? What are those chambers doing? Oh, okay. Be very careful. The word pumping is for the whole heart. When I specify the chamber, contract. Contract versus relax. Thank you for not saying expanding. Okay, a very common error there. So LA pressure smaller, LA relax. LV pressure greater, LV. And be very careful though. We can only use this for chambers. For example, aortic valve is between the aorta and the, look at your diagram there. Aortic valve, LV and aorta, isn't it? Okay, so if I say that the aortic valve closes, for example, this one, I could still leave it as LV, but this one becomes aorta. If the aortic valve closes, pressure is this way. Okay, understand what I'm doing here first? Yes? But what is the aorta? It's a vessel. It's a vessel. vessels do not contract or relax. So we write nothing here. Only chambers contract and relax. Okay? So in your explanation, when it's an open-ended one, please be very careful. We only write down the contract relaxing if it's a chamber. If it's a vessel, say nothing. Yes. Muscles of the LV. Muscles of the, yeah. I think it's a small thing though. Got marked down. Anybody get marked down for saying the chamber contract and relax instead of the muscles of the chamber? Is that a hand raise? Oh yes. Wow. Okay. Can. That's good information. Thanks for letting me know. Alrighty, let's go. So, last but not least, when we learn about things, a very common theme across topics in bio is what happens if it goes wrong. So coronary heart disease is where we look at our coronary arteries, and it's where just now we mentioned, cannot say heart to heart, coronary arteries. The coronary arteries transport oxy blood from the aorta to the heart muscles, right? From the outside. Okay, from the outside. Why would they become narrow or blocked? Why would they become narrow or blocked? The build-up of two things. Fats and egg yolk have a lot. Prawn head have a lot. Cholesterol, thank you. So we're looking at this. Okay, so coronary arteries may become narrow or become blocked. But hey, if you look at this could happen to any blood vessel. This could happen to any blood vessel, isn't it? So why are we focusing on the coronary artery? Number one, they're pretty small for an artery. So the smaller the lumen, the easier it becomes blocked, isn't it? That's number one. Number two, this one is to the heart muscle. The moment this one become blocked, your heart muscle how? Correct? It doesn't explode, thankfully not immediately. What we need to think of is, of the many things that blood transports, we want to point out two. Glucose and oxygen. Thank you. When we talk about glucose and oxygen, why do the cells need glucose and oxygen? Process is aerobic respiration to release energy. So when we come to this point-to-point analysis here, if the arteries are blocked, less oxygen and glucose, less aerobic respiration, less energy. The moment you get to energy, then you can say the cells die. Please do not jump from less blood die. Okay, we will never do that in our syllabus. Never less blood die. Can always tell me about what's in blood. Less blood, less oxygen and glucose, less aerobic respiration, insufficient energy. Then the cells start to die. Okay, and this is something that applies to all organs. Applies to all organs. Lauren, cannot open, cannot eat. must have skill one. Okay. George asks, dilation and constriction versus contraction and relaxation. When do we use dilation and constriction? When do we use dilation and constriction, people? What dilates? What constricts? Not the entire eye. I'll take that. The pupil, right? Okay, the pupil, right? So, we learn to use the terms dilation and constriction in the topic of the human eye, right? Pupil reflex, for those who have learned it. But generally, what does the word dilation versus constriction refer to? A narrowing or a widening of a circular space. Okay? So we can actually use constriction and dilation in the topic of digestive system. What constricts and dilates? In a wave-like fashion. Peristalsis, thank you. So peristalsis is the alternating dilation constriction law, right? In the esophagus and in the esophagus. All right. So, George, are we clear on that now? Thanks for asking. Come, let's move on. Respiration, yes. Okay, respiration. So fairly straightforward, fairly straightforward stuff over here. Nothing too much. We have all learned this before already. What I want to jump straight into is the trachea. I want to look at mucus producing cells and the ciliated epithelial cells. Okay? So I've highlighted the cells in purple. I've highlighted the working components in green. Now, a very common error is found here. May I ask you all now, think of what the commonly wrong answer is. This part that I've boxed up in orange. What's a very common error that people write down when we talk about the function of cilia? No? The pushes mucus part. What's the error? Usually, very good. Please keep in mind, cilia does not push dust away. How do you describe cilia? It's just a hair-like structure, isn't it? So I ask you all, later go home, go and take the broom. Like that. You're not going to be able to sweep dust away, isn't it? It's just going to float back and forth in the air. Right? So the mucus must trap the dust first, then you sweep the mucus away. Right? A very common error is cilia sweeps dust. So in terms of description, how can we best optimize our explanation so that our words, our sentences don't get too dangerously long? Talk about mucus first. Mucus trap first, then easier, your next sentence just say cilia sweeps the dust, cilia sweeps the mucus away. Okay? Right? So please remember that, right? Pushes mucus, not dust. Next thing I want to look at here. Where? Direction is very important. Obviously, you're bringing it away from the lungs. That's what I'm trying to protect, right? Where is it going to? It's going to the throat. And yes, you read that correctly. It is to be swallowed or expelled. Right this very moment, none of us are too sick. So therefore, we are swallowing mucus goodness. It is going right now. Down, just swallow your saliva. Mucus. Why is that fine? Why is that fine? Link to digestive system. When you swallow something, where does it go? Through the esophagus into the stomach. It is the stomach where we have our answer. Why is it okay to swallow the mucus there? What is in the gastric juice? Hydrochloric acid. Yes. But we need to specify hydrochloric acid. And at this point, the moment you specify hydrochloric acid, okay, really typically most of the pathogens that you swallowed in mucus there will be destroyed by hydrochloric acid. Okay? Right? It is only when there is excessive mucus production, then you have to then. Okay? So that is when there is excessive mucus production. What stimulates excessive mucus production then? Okay, excessive dust. It doesn't have to be dust, it's just something that irritates your airways. Okay? Right? So it could be a physical irritant like dust, or it could be a pathogen, right? It's something that's infecting, damaging the cells there. Clear? Alright, we'll talk about that later on as well. Come, let's move on. Gaseous exchange, easy stuff. Learn this since primary school already. Right? What I want to draw over here is this. And identify for me. Annabelle, what is this purple thing that I'm drawing over here? No? Thank you, the thin film of moisture. And I have left that in the table below as well. Look for thin film of moisture. You see it there in the table? Thin film of moisture. And I want you to highlight the two D words. Dissolve, diffuse. Now, very importantly, we need to have that dissolve then diffuse because there are no gaseous gases in the body. Do you understand that? Gaseous gases? Gaseous gases. The gases are all in the what state? Aqueous state. Thank you. No gaseous. When they dissolve, they become aqueous. Okay? Right? There should not be any gaseous gases in the body, except for the digestive system, for example, and that's where gases escape, right? So in the bloodstream, it's all dissolved. Right? So therefore, before that absorption of oxygen, it will dissolve first, and then it can diffuse. Okay. All right, and then again, bring your attention back to the whole idea of movement of substances. Surface area to volume ratio, diffusion distance, surface area to volume ratio, steam concentration gradient, diffusion distance. Again, the topic of movement of substances emerges so importantly. Okay? Alright, so please make sure you go back and revise that. Let's go. Breathing, respiration. Which one can we control? Anybody can control respiration? Oh, that will be a miracle, okay? So we can control our breathing only, but we are limited still by respiration. Okay, right? So be very aware of that. Breathing is simply how air moves in and out of the lungs. That's all. Okay, and then what is respiration? Releases energy. And it is at this point here, I want to make sure it's on the top of everybody's heads here. Purpose of respiration is always to release energy. All right? Sometimes we can be so used to learning about this in primary school. Oh, what does respiration release? Carbon dioxide and water. Then your first answer to me is carbon dioxide. Is carbon dioxide produced? Yes, but that's not the purpose of respiration. Our primary thought in our head should always be energy first. Okay? Before we look at respiration and the rest of the page, let's talk about breathing a little bit. We have to learn about the process of breathing. The process of breathing. Can I use the space over here at the bottom? So breathing. Anyone recognize this? Who's Eric? Eric is my friend that only exhales. He never inhales. Right? We're going to use this to remind ourselves that ventilation, the breathing process. Right? So I typically use Eric exhales and then you just flip it around because Rice inhales doesn't quite make sense. Okay? So Eric exhales. This is a person exhales. What does ERIC stand for? I bring your attention to E and I. What are the Laura, help me close the door, please. The door no spring on, must close properly. Thank you. No problem. Your ribs are connected to muscles. The external and internal what? Intercostal muscles. Now, this one, don't write. Just look on screen first. Come on, people. 1 and 2. Which one is the correct spelling? 1. Thank you. Intercostal, not intercoastal. Alright, there's no A there. It is not coast to coast, not coast to coast, it's intercostal. C-O-A-S-T versus C-O-S-T. You see that? Right? But E and I, another error is people start to say external costal, internal costal. The name of the muscle is intercostal. There are 2. So if I ask you to look at my fingers over here, I'm going to use this as my ribs. Okay, these are my ribs like this. If I open it out like that and show you, the muscles here are the external intercostal muscles. The muscles here are the internal intercostal muscles. Okay? So on my ribs here, external, internal. Can you see that? Right? So, Eric, like this. ER. So the first thing we think of is here simply it is a mouthful, but do your due diligence to get the marks. The external intercostal muscle relaxes while the internal intercostal muscle contracts. Now, at this point, the muscles are connected to the ribs, to the entire rib cage. So, muscular movement results in skeletal action. At this point, don't memorize. Just exhale now. What your rib cage do? Exhale, then do what? Your body's going to sink downwards, right? Then say downwards lah. The rib cage moves not just down, it's going to move from the sides. If you look at my hands here, inwards as well. That's it, right? So this tells me the rib cage moves downwards and inwards. But there's a third set of muscle that we need to include here. The diaphragm. And at this point here, how do I spell the diaphragm? How do I spell the diaphragm? Let's hear from Trisha. Well done. The diaphragm, right? Right? So it is spelled diaphragm like this, but it's going to be pronounced diaphragm, right? The diaphragm, what happens? Tag the diaphragm to this. Whatever the external does, the diaphragm does as well. Okay? Alright, so therefore, diaphragm relaxes. What's the effect of that? The relaxation? What's the effect of the relaxation? Okay, so there's something I use as well. So over here, very simple. We're gonna have the diaphragm right at the low rib there. Right at the bottom of the low rib here. Think about it. When you exhale, do you want more or less space in your chest cavity? When you exhale, less. So therefore, your diaphragm is going to relax and curve upwards. So look at my hands here. When I'm gonna take a breath in, look at my hand here. This is my diaphragm. It's gonna push the air out. Pull the air in, push the air out. Can? Alright? So therefore, this is something that I'm doing to show you. So when you are doing your exams as well, don't go and do it subtly like at the bottom of your table here, but that is how it works here. Think of yourself, the diaphragm pushing the air up when you exhale, pulling the air in. Okay? Alright? So diaphragm relaxes and curves upwards. So at this point, the combined action of the rib cage and the diaphragm causes the, what do I refer to this? What did I refer to this just now? Either the thoracic cavity or the chest cavity, both work. Okay, what happens now? Exhale, exhale. The space, you want more or less space? Exhale, more or less space in the lungs. Less space, but less space. A bit too not scientific, right? Volume, thank you. Chest cavity. Decreases. Chest cavity volume decreases. But when volume decreases, something else comes into play. Pressure, very good. So, I'm drawing arrows over here because it is linked. It is a cause and effect thing. When volume decreases, can I ask you to think about the space in this classroom then? When I make this classroom a little bit smaller, all of you are going to feel a little bit more squeezed. And that is pressure increasing. Can? But so what? Here is where I want you all to bring up an idea about homeostasis. Have you learned homeostasis yet? Or even without homeostasis, a seesaw. You know how seesaw works, right? The other side must be heavier, then I will go up. If I'm the heavier one, then I will do this. So we're always doing a comparison. What am I comparing to? Surrounding and atmospheric. Very good. When pressure increases, you cannot stop there. When pressure increases, atmospheric or surrounding both work. Okay? Alright, when pressure increases to above atmospheric, this is where we have acceleration because air also moves from higher pressure to lower pressure. So when we get here, you can see. For those who are done transferring this down to your handout, right? Think of the opposite then. What are the words you will change if you are asked to talk about inhalation now? Look at what you've written down. Don't look at my screen. Right? For the most part, the opposites are very straightforward. Okay, if you're talking about inhalation, just flip these two. Out and up. Isn't it? Contracts. But here is where our first caution appears. Just based on the words, please do not say curve downwards. Flattens. Why? Where? You know where your diaphragms are? What's below your diaphragm? Your liver. Your stomach. Hello, every time you breathe in, then everything squash down. Why? Every time I go toilet, no. Okay? So therefore, it doesn't move downwards. It simply flattens. And the reason why I use my hand, right, is because I cannot make my hand the other way. I can do this, I can flatten, but I cannot. I mean, I can ask like that, but then that doesn't happen. Okay, so curve up, flatten. Okay, I'm not gonna like form like an inverse of V like that. Okay. Right. Okay, right. But other than that, really, this is the only thing that I want to bring to everyone's attention. Do not say curve downwards or moves downwards worse. Okay, that's it. Right, so the only thing I'm looking at here is instead of curving downwards for the opposite for inhalation is flattens only, just flattens. Alright, so that's for breathing, inhalation, exhalation. Let's head back out over here. Respiration, like I said, respiration, primary focus is by energy. In this case, I've written just the words energy here. What's the difference in the energy? Large and small, thank you. Right, which one is the large one? So please remember, aerobic respiration is always the primary type of cellular respiration. Okay? Okay. Aerobic, anaerobic. First difference that appears in our head is presence of oxygen. Okay? Second difference is the energy production release, that one we talked about. There's a third one. What's the third difference between aerobic and anaerobic respiration? The products, right? Okay, in aerobic respiration, relatively harmless. Carbon dioxide, just exhale. Water, you use or you sweat or you pee it out. Fairly harmless, isn't it? But lactic acid. How? Lactic acid is toxic at higher concentrations. So what do you do with lactic acid then? What do you do with lactic acid then? Oxygen. This is where oxygen debt comes up, isn't it? Right? Oxygen debt comes up. So therefore, how do we deal with lactic acid? Let's go on to the next page. We are looking at lactic acid and the oxygen debt. And yes, you heard me correctly. It is spelled with the B but pronounced without. It is oxygen debt. All right. So what is the oxygen debt? Actually, the term oxygen debt here, not as important as what is it. So what is the oxygen debt? Highlight for me. So, questions here, we have it appearing as lactic acid is found in the muscles, isn't it? After the muscles undergo anaerobic respiration. What is the first thing that happens? We want to talk about what's the first thing that happens to lactic acid after anaerobic respiration? A very common answer is broken down in the liver, isn't it? But where is it produced at? Muscles. Then from muscle, we suddenly jump to breakdown at the liver. We have to bring in the transport first. We have to bring the transport. So there's a very commonly missed out point here. Lactic acid in the muscle first. Transport in the blood to liver. Then you can break down in the liver. Alright, so please remember that. Please remember that. Why does anaerobic respiration occur? It's not about glucose. Plenty of glucose. Anaerobic respiration occurs when there's not enough oxygen. Oxygen. Not enough oxygen to continue the high rate of aerobic. So when anaerobic respiration occurs in the muscles, what happens to aerobic? What happens to aerobic? Trisha? When anaerobic respiration occurs, what happens to aerobic respiration? All living cells undergo aerobic respiration all the time. Exactly. If this stops, you die. Never. Aerobic respiration never stops. Please keep that in mind. Please keep that in mind. Annabelle, help me out then. What happens to aerobic when anaerobic respiration occurs? At what value? Thank you. When anaerobic respiration occurs, aerobic respiration maintains at whatever that maximum value was. The whole purpose of anaerobic is your aerobic cannot increase anymore. But I still need more energy. That's why anaerobic also occurs. Use this very simple word, also. anaerobic respiration also occurs, right? And the moment we use also, okay, that means both are happening. Can? So please remember, okay, done with lactic acid. Then let's move on. Harmful effects of tobacco smoke. There are plenty of substances in tobacco smoke, cigarette smoke. Our syllabus only requires us to learn about these three. So for each of them, I'm just going to very briefly share with you. I'm just going to highlight a couple of words here. For nicotine, can I ask you all to remember causes addiction? Number one. Number two, increases heart rate and blood pressure. Okay? Nicotine, that's all. For tar, what does it mean to be carcinogenic? Cancerous, cancer causing. Thank you, I like that. Number two, paralyzes cilia. What is cilia supposed to do? The sweeping action, isn't it? So now with tar, what happens? It's gonna get paralyzed. It cannot sweep anymore. But what do the airways do? Supposed to sweep, but the mucus stays there. So, but the tar is still there. So I produce more mucus to try and remove it. But the cilia cannot move, so I produce even more mucus. Eventually, your airways become more and more blocked. Right? Okay, more and more blocked. And if, unfortunately, because the cilia cannot sweep it away from the lungs, tar makes its way into the alveoli. And this is what a smoker's lung looks like. Not only is this an aesthetic change, right? It just becomes black. But I mean, it's in your body, right? Black, black lah. But the idea here is, please take note, because it's sticky, what are our lungs supposed to do? We breathe. Our lungs. What do balloons do? Inflate and deflate. Thank you. The lungs are elastic tissues, but because the sticky tar hardens the tissues, it can no longer deflate and inflate as efficiently as it should. So that's why smokers tend to have higher resting breathing rate. They need to breathe faster because they cannot breathe as deep as before. Okay? So thanks for asking. All right. Last but not least, we talked about this just now. We can combine more readily and more strongly into just one word, isn't it? What is that one word there? Carbon monoxide binds irreversibly or permanently. Thank you. Okay? Right? So with every hemoglobin that does that, we lose the hemoglobin's ability to transport oxygen, less oxygen. Last but not least, we talked about it just now. Here we see the diagram. What is plaque made of? Fats and cholesterol. Wonderful. This plaque is different from the plaque in the teeth one, different. So take note of the context. Plaque in the blood vessels, fat and cholesterol deposits. Okay? So this is what we're looking at. We're done. Let's go. Excretion. First and foremost, I placed in there the definition of excretion. Can I ask you all to look at this definition here? Is there a difference to what you learned in school? There is a slight difference here. Kelly, what's your difference? Sorry? Yes, what is missing in this definition here that you learned in school? Thank you. The first two are actually the same thing. We are just going to combine it as such. Toxic metabolic waste, isn't it? Right? What I want to ask us to write down together here with me, excretion is the removal of toxic metabolic waste products. And substances in excess. All right, so the evolution of the syllabus has resulted in this idea being brought in as well, because the most important mode of excretion is urine. And here we are looking at within urine. I mean, there's water in it. That's the primary substance in urine. Is urine a toxic metabolic waste? That's what I'm saying. Is water a toxic metabolic waste? Absolutely not. Water is important, it's necessary. So then how? Why is it still found there? Excess law. Can? Excess. Right? So I've put in over here. I mean, definitely we understand that this topic in our syllabus focuses on the kidney, right? But I also want to make sure that we all know that these are the other organs that are involved in excretion. The organ of excretion is the lungs. What do the lungs excrete? Carbon dioxide. Very good. What does the skin excrete? Method is sweat, right? We got very, very, very careful. So you look at the okay, no. So over here, right, sweat, we have water, you have salts. Right? So this is what we refer to as the excess thing. Can? Liver and large intestine is an interesting one. What are they excreting? Urea is for kidney, isn't it? Liver and large intestine, how? So this is where we must be very clear. Excretion versus, thank you, Fiona. Excretion versus egestion. What is egestion? Removal of what? Thank you. Undigested food. From here, we immediately make a contrast. Undigested food versus just the word products. Products means your body make one But undigested food do you make the food in your body? It's just simply your body cannot do anything to it. Very, very big difference. So feces primarily contains undigested food. What else does it also contain? Yeah. We are looking at where does bile come from? Produced by the liver, thank you. Stored in the gallbladder. Released into the duodenum, thank you. Because we learned the parts already, we cannot state small intestine, must state duodenum. Okay? Right? So once more, produced in the liver, stored in the gallbladder, released into the duodenum. Because of that, released into the duodenum, meaning it will move along together all the way. And therefore, it does not get reabsorbed and hence excreted with feces. So bile salts is a mixture. The part that emulsifies fat, that one we need it. We use it lah. But within the mixture, we have excretory products of bile as well. And that's why it's going to be excreted. Okay? But no worries, our syllabus simply focuses on kidneys and urine. Carbon monoxide binds irreversibly. Okay. Why must there be deamination? We talked about that already. Why must there be deamination? Why is there no such parallel for carbohydrates? When we consume excess carbohydrates, what do we do with it? Glycogen, yes. Glycogen, but we have no such parallel for proteins and amino acids. So we use whatever we need to, cannot store. So if you use, you cannot store, break down. And this is deamination. As the word suggests, deaminate, remove amino group. Okay? For our syllabus right now, that's all. We do not need to go deeper. Okay? To deaminate is to remove the amino group. We don't have to go deeper. That's all. But if you want to, you can have a look over here. Right? I've put it in our diagram here like that. And then a throwback into past topics here. Blood vessels. Please make sure we remember the names, isn't it? Right? Organ associations, hepatic, renal. These all where it all comes to play. All right, please make sure we are very clear about that. So what are the two processes that happen in the kidneys? In order. First process is ultrafiltration. Second one is selective reabsorption. Now, typically, we can also learn the third process that ties into homeostasis. What would that be? Osmoregulation. Thank you. What is osmoregulation? To maintain something. To maintain what? Water potential of blood. So just maintain blood water potential. Okay? So that's three processes like this. Structure of the kidney, I can leave it for you to look at. What I want to focus on here will be simply the middle segment here. The renal artery, vein, and ureter. Right? So sometimes, obviously your papers will not be colored in your exams, isn't it? So how do we identify the renal artery and renal vein? If they never give you color, they must give you either one of these two things. Arrow. If the arrow direction of blood flow, then you identify it, isn't it? Okay. But if they never give me color, never give me arrow, what is the only other thing they have to give me? Thickness of what? If you say thick, then it's not lumen already. Thickness of the wall, isn't it? The vessel wall. But if you want to say lumen, then it's diameter, how wide the lumen is. And then this brings us back to just simply artery versus vein. Can? So link your ideas together. You will never get colors in your questions. So you depend on arrow or blood flow or vessel structure. Either the wall thickness or lumen diameter. Can? Use that to identify. As to why you will never get colors, because it's expensive to print. Okay? All right. And then usually the blood vessels will be either going up or going together. The one that is the odd one out will be the ureter. Okay, please make sure, you must be very careful. Ureter is not to be confused with the urethra. It is not to be confused with the uterus. Okay, very similar sounding and spelled words. Okay? Okay, come, let's move on to the nephron. Now we have this entire mass of tubules like that, isn't it? This entire mass here. Right, I'm going to use this space here at the top. Just draw a very quick sketch, right? See first, don't draw first, That's it. This is essentially the entire nephron. Make it simple. Make it simple. Your entire nephron is just scribbled like this. Okay? Part by part, come. Ashley, what's this? Bowman's capsule. Yushen, the next one is? Proximal convoluted tubule. Thank you. Shalise. Did I say your name right? Shalise? Shalise? Hi, Shalise. What's this? The loop of Henle. And at this point here, loop of Henle must be uppercase H. It is named after a person. Can? No need to do the accented E. But the uppercase H must be there. Can? Zihan. The distal convoluted tubule. Thank you. Reina? Wait, you not have the material? Okay. What's that? Distal convoluted is here already. Proximal is here already. The last part of the nephron. Collecting duct. Thank you. Okay, so keep it simple. Now, I want us to understand proximal versus distal convoluted tubule. Tubule because it's just tube, very simple. Convoluted because it's in a mass. So proximal versus distal. Anatomical terms for near and far. Proximal near, distal far. What is it in reference to? Near to what? Far from what? The Bowman's capsule. It is always with reference to the Bowman's capsule. But not like location, but more of the fluid movement. The fluid that is produced from here, what do I call that? What's the process that happens here? Ultrafiltration, right? Then chemistry, we learn already, the fluid that comes out from filtration is called filtrate. What is this thing called? No, I'm drawing the glomerulus, thank you. So therefore, the fluid that is produced via ultrafiltration is known as the glomerular filtrate. So the identification of the PCT and the DCT is based on movement of the filtrate. From the Bowman's capsule, which tubule does the filtrate reach first? That one proximal lah. Then you flow, next one, distal. Because why? Sometimes I've seen a very cunning diagram drawn this way. If you base just on location, then this one look nearer, isn't it? Right? So it's not based on location, but more of movement of fluid. Okay? The first convoluted tubule after the Bowman's is proximal. Next one, distal lah. Okay? And literally, if you can remember just this diagram, you are done. You are good with your nephron already. Right? Don't need to worry too much about the other structures. Clear? So it's all like that. Okay, now this one. Grammatically, people, both are afferent arterioles. Afferent and efferent. But it's going to be very difficult for us to learn if I do that. So I'm going to say afferent and efferent. Okay? And I'm going to do the same thing as I did with transport in humans just now. The alphabet starts with letter A. Our journey begins with the afferent arteriole and leaves by the efferent arteriole. Okay? Alright, so blood flows this way from A to E. Always from A. From afferent to efferent. So what's the difference over here? We are going to bring in what Sonny brought up just now about the arterioles. And my diagram actually shows a little bit about this. Can we see the difference? My diagram is going to be clearer. You can see it here. Lumen diameter. So it's not about thickness. Okay. So the afferent wider, efferent narrower. Very simple idea here. Think about your entire cohort after assembly. From the hall, you all now must filter out into the narrower corridors, right? But you're very nice people. You all let people through. You don't squeeze. But imagine now, everybody must walk at the same pace. What increases? Pressure. And that's it. And that's actually the meaning of the ultra, behind the ultrafiltration. There is pressure involved. If I just say filtration, then it's just gravity-led, Okay? So ultrafiltration tells me pressure is involved. Right? If pressure is involved, I will use the word force. Forcing what out from the blood? During ultrafiltration, what is forced out of the blood? What do I force out of the blood? To form the filtrate, what am I forcing out? Things in plasma. So over here, the distinction is size. Ultrafiltration results in the forcing of the ultrafiltration results in water and small solutes being forced out of the blood into the Bowman's capsule. And this forms glomerular filtrate. Okay? But if you look at my screen over here, which one is the outlier? Urea. Why? Why is it outlier here? Why? Why have I placed urea in a different color from the others? Exactly. Keep it simple. Good stuff, bad stuff. Everything over here, urea is the only thing that we consider bad stuff. And therefore, in the next process, after ultrafiltration comes, selective reabsorption, I will not selectively reabsorb any urea at all. That's it. Okay? For our syllabus, the primary focus is on all glucose and amino acids to be reabsorbed. Okay? Common questions are like, why are there no blood cells in the urine? To pass through which stage? Ultrafiltration. Very good. So ultrafiltration, look at the word I wrote down, small. Anything that's too large will remain in blood. Okay? Next question then. Why is there typically no glucose in urine? Come out what? Yes, it is selectively reabsorbed. We just highlighted that, isn't it? Okay? Right? So one explanation is too big, doesn't even go out. Another explanation is does go out, but selectively reabsorbed. Okay? So this is what should typically happen. So then issues can arise. Hey, what if there is blood cells in the urine? Where is the error? Where is the damage at? Cannot say kidney. Where is the damage at if there is blood cell in the urine? The glomerulus or the Bowman's capsule. Okay? Right? Always think about that, Okay, last part over here, we have osmoregulation. This is where we have the overlap with homeostasis, as I mentioned earlier on. Please take note. Always reference the norm. Always reference the norm. The norm refers to a range of values. A range of values. Okay? Range of values are. So I believe that in how I have put this diagram together, it is going to be very sequential. So look at it from point to point. Hypothalamus and pituitary gland would be relatively newer terms that we need to be comfortable looking at and identifying. Right? Hypothalamus and the pituitary gland are both found near the base of the brain. Right? The words here are very important. Detects is stimulated. So the hypothalamus is the one that detects, whether or not blood water potential has changed drastically enough. The pituitary gland is stimulated. So who stimulates the pituitary then? Hypothalamus. Hypothalamus sees the change, tells the pituitary, release more, release less. Okay? What is the function of ADH? Firstly, what's ADH? Thank you, antidiuretic hormone. So the function of ADH here. The things I've highlighted here, we're just going to clump it together. Versus. So what is ADH for? Of the collecting duct. Thank you. All right. So ADH regulates the permeability of the cells of the collecting duct. More ADH, more permeable. Less ADH, less permeable. That's it. That's it. And keep it simple. And so if previously above the norm, now because I reabsorb less, then my blood water potential how? From just now above, now back to the norm already. Do I still need all these effects? I don't need any more. Hence, Previously, I decreased. Now I'm going to increase it back to the normal levels. Done. So therefore, it is a constant back and forth. Too low, I bring you up. Too high, I bring you down. Keep on balancing. Okay? And the last part over here, similar as what we have talked about in transplant in humans, coronary heart diseases, isn't it? Over here, we're looking at kidney failure and dialysis. Now, very important, dialysis is only required if the person unfortunately has total kidney failure, meaning both kidneys are damaged. Because our kidneys are extremely hyper efficient and well engineered. In fact, right now, both of our kidneys are not functioning at like half even. It's not like I have two, okay, like half. No. 30-30, more than enough for our users. And that is why the kidney is one of those organs that can be voluntarily donated from a live donor. Alright, okay, so the kidneys are very great at their jobs. I put in many of the functions over here, right? But most importantly, it's about nitrogenous waste, the removal of it. And over here you can see what does it remind us of again? Movement of substances. Okay, so time and again, the topic appears so many times. And not to worry, dialysate is just another word for dialysis fluid. Okay, no need to worry about it. And with that, we're done. Okay, we're done. All right, so we have a couple of questions given to you over here. Okay, now the point here is that the answer key will be provided afterwards. So I do not need you all to walk through every single word in the open-ended, especially later on. But may I just challenge you all to just take 10 quick minutes, try the 10 MCQs here first. Okay, if you need to go to the washroom, please go ahead on your own right now. We'll come back together after 10 minutes. We're going to look at the questions together here. Okay, so for those that are still sitting here with me, can try the questions first. Did you want the complete? So sorry. Did I miss out? Possibly.
I already
Ah. Maybe about 2 more minutes, we're gonna come back together and look at the question, huh? Picnic Picnic Why don't you help me close the door, please? Thank you. Natalie, yes, we are looking at the red blood cell. When we talk about donations, we focus on the red blood cell because plasma is just soup. No need to donate soup. The red blood cell is more important. Can? Okay, let's go. Let's come back together. Right. For those who went for a break, I hope you are feeling a bit more refreshed now. For those who have been trying the questions, let's get on with it. Let's identify some of the structures here first. Right. Let's identify some of the structures here first. Anushia. D. Pointing to nothing, isn't it? But this is blood smear. When you okay, the same idea goes when you have a cell, then this is your nucleus, then your whatever, then the label is here. What's this? Yeah lah, then if blood leh. Oh my goodness, hey, don't destroy door Cytoplasm of blood. Yeah lah, plasma.
Sorry.
Okay please take note This nothing is plasma. Okay? Lauren, help me out. B and C, choose one. What's that? How do you know that? Very good. Look like the drawing that I drew, right? That's why. Okay? So the lobed nucleus of the phagocyte is what is going to help us to identify the phagocyte. Okay? Nico, what must B be then? The lymphocyte. How do you know that? The humongous nucleus. Thank you. What's A? We already left it A. What's A? It's the red blood cell. Right? Take note the red blood cell, what are the words we use for the description of the shape? Biconcave. Biconcave means the middle segment is thinner, isn't it? What instrument are we using for this diagram to appear?
Microscope.
Light microscope. We're using a light microscope to observe this, meaning if the middle is thinner, more light can pass through. That is why the center brighter. Alright? We learn to pick up these small little skills to help us understand diagrams better. Biconcave shape of the red blood cell, center thinner, more light can pass through. That's why the center is brighter. A is the red blood cell. And as the name suggests, phagocytosis over here. What are the two words for phagocytes? Function. Number one is what? Engulf. Thank you. Number two is then breakdown. Wonderful. All right. Engulf and breakdown is phagocytosis. Let's move on. Antibodies. Is it on the RBC or in the plasma? Plasma. Thank you, Annabelle. Right. So over here, very simple. These are the things that are found on the. Oh, where's my highlighter? There it is. So then when we look at the donor, the donor's blood. What do they have then? If agglutination occurred over here, what does it tell me donor P has? A what? Antigen A on the red blood cell. Thank you. Okay. So P tells me, so over here we're gonna do another column ourselves. So this tells me we have A. Right? And then look at the next column. No agglutination with B. So got B or not? This P don't have lah. So A only. And let's do the same loh, like that. How? Agglutination observed as well. So A is present. But next one, also agglutination observed, meaning B. So based on this, we therefore know that donor P is blood type A. Based on this, donor Q is AB. Can? But the question is not asking us to find this out. The question is asking us, these people can donate to who? From here, we've done so much work to analyze, but actually we can answer the question without analysis. Why? Thank you. Universal recipient. Okay? Alright, but the thing is, we may not be always so lucky. So we still need to learn how to analyze the question. Can? Let's go. Ray asks, donor P has antigen B? Huh? No, this is a test. This is a test. We did a test by adding these antibodies to their bloods. Ray, you misunderstood the first table here. Is it clear now? Okay. Okay, graph. What did I ask us to look at immediately? Intersections. Thank you, Chloe. Right, so look at intersections only. Here they're asking us open and close. So firstly, bicuspid. Release down. Bicuspid. Nicole. Bicuspid between where and where? Between? Flip lah, flip lah. The bicuspid valves is found between what two regions? Very good. Okay, bicuspid is found within left atrium and left ventricle. Alright, so we just choose any one. This one, left atrium, left ventricle. This is one intersection here. This is one intersection over here. So at this intersection, LA greater or LV greater? That first intersection, LV or LA greater pressure? LV greater pressure. We do this. We put a P. We ask ourselves, correct or wrong direction of flow? Wrong direction. So the valve closes. Okay? So with that in mind, question is asking us to just look at X only actually. So if here the bicuspid closes, the next intersection is here, isn't it? This is when it opens. Time X is before or after the opening? Before, so therefore it still is closed. Alright, so graph skills are, please open your big beautiful eyes, look at the graph carefully. Here, close. Here, then open. But X is before the opening, so it's still closed. Our answer, we have to choose close. And let's apply the same thing. Time X is here. Let's look at this. This intersection, what is happening? What's happening over here? This one is between LV and aorta. What's the difference in the pressure? Whose pressure is becoming greater at this intersection? Aorta, thank you. We just put our pressure here. That's it. So look at this arrow. Correct or wrong direction? Wrong. So the valve closes, exactly. So therefore, the aortic valve, answer is A. Can? Let's go. So, breathe in. When the diaphragm contracts, what's the action? Contracts and flatten or curve up? Contract and flatten or curve up? Flatten, thank you. When the diaphragm flattens, are you inhaling or exhaling? To flatten is to inhale. So here we're talking about inhalation. Question gives us volume and pressure. So if it's for inhalation, I'm trying to decrease pressure. So we're looking at pressure needs to decrease. But the question says starting to. How? Starting to. C, right? At the start, at the crest. At the crest here. Right, we are starting to inhale. So therefore, here starts to decrease already. The next cycle happens again here. This is where you have the next increase then in the volume because inhalation occurs. Can? So the word starting to is important. The word starting to is important. Okay? For question 5, not just athletes, normal folk like you and I, after a race, after running for the bus, after your 2.4, after you have stopped running, stay still. The breathing still remains very high. What's that for? For the oxygen debt, isn't it? But again, the oxygen debt doesn't help us to explain why does our breathing remain high. We're talking about breaking down lactic acid. Right, we have some requests for 3. Hold on. Bicuspid open at X. Clarice, if you focus carefully, X is here. The intersection is here. It opens here. So close all the way. Can Clarice? Can you see the graph properly, Clarice? All right. What's the another question? Was there another question? Okay, and then for 4, looking at starting to. So when it's starting to contract, the pressure will not have changed yet. So therefore, it is right before the decrease in pressure. Right before the decrease in pressure. Right, so our answer is C. Okay, moving on. Question 6. That's fairly straightforward one. This one. This is just about harmful effects of cigarette smoke or tobacco smoke. Right. Even without the last column, we should be able to find some ideas here. For nicotine, what is the word we are looking for? What is the addiction? Thank you. For tar? Paralyzed. Very good. And there we have it, isn't it? Paralyzers. Okay. So actually on this note here, emphysema and bronchitis, they are actually errors in different parts of the lung. What is emphysema? What is emphysema? What part of the lung is damaged? The person is suffering from emphysema. Emphysema is due to chronic and excessive violent coughing. What gets damaged? The alveoli. The alveoli are the tiny little balloon-like structures, isn't it? Right? But due to excessive coughing, the balloons burst. So instead of having many tiny alveoli, we have a few moderately sized ones. Why is that a bad thing? Surface area to volume ratio. So tell ourselves, for emphysema, it is damaged alveolar walls. Bronchitis. Anything itis is inflammation, infection of. So what's inflamed? Bronchitis. Not the trachea, but the bronchi. Right? The bronchi. Right? So here is where we have the bronchi. Kate asks, why is it need A? It's not A here because I can see you're looking at volume increasing, therefore inhalation, but we are looking at pressure changes as well. Okay, for the diaphragm starting to contract means haven't inhaled yet. Pressure should not be at this point so low here, right? Pressure should start to decrease when the diaphragm is contracting. Pressure needs to start to decrease, so that's over here. Okay, Ken? Okay, let's move on. We talked about 7 already earlier on about carbon monoxide. Question is asking, why does carbon monoxide poisoning result in such symptoms? From the 4 points over here, which one have you talked about that carbon monoxide does to our body? Which one are we looking at? Hemoglobin. Answer is B. That's it. Okay? The trick here is actually statement A is true. Statement A is a true statement, but it doesn't help with the explanation. Gotta see that, MCQs, you must choose factual and relevant answers. So A and B are factual. Which one is relevant? One is B. Then for the breathing portion. 6 is D, Cayenne. Emphysema and paralyzes cilia. Okay. All right. X, Y, and Z, fairly straightforward. Urea produced in X, filtered in Y, stored in Z. Like this, you chose? Choose now. You choose what? Not Annabelle choose one. Yes, it is our answer, yeah. Okay, right. Alcohol consumption reduces the production of ADH. So more alcohol means less ADH. Question is asking, alcohol does what to the amount of water reabsorbed? Alcohol means less ADH. So work from there. Less ADH means how much water reabsorbed. Is it more or less? Less lor. So therefore, 10. So then how? Urine production how? If I reabsorb less water into the bloodstream, more water remains in the filtrate. What is the fate of filtrate? What does filtrate eventually become? What's the name of that fluid there? What does filtrate? If filtrate becomes urine. So if I reabsorb less water from filtrate, more water remains in filtrate, more water in urine. Quantity increase. And you think about your Milo lah, more water diluted. Okay? Question A is A. Alright, last one. This is what I was cluing everybody into just now. T, U, and V. Can we identify them first? So I purposely grayscale this picture. No color for you. No color, no arrow. What do we do? The lumen, isn't it? Is it obvious enough? Kinda, right? Kinda. So we have T as the one that has a narrower, it's slightly smaller, but also this is where I can share with you something else that you may not have been exposed to in school. The arteries means away from the heart to the kidney, right? When it goes to the kidney, what's the first thing that happens to blood? What happens to blood in the kidneys? Ultrafiltration. Which part of the kidney does ultrafiltration occur at? Which part of the kidney not the nephron. That's nephron already. Region of the kidney. Cortex, thank you. So therefore, I left it in your notes. I left it in your notes. By the way, if you all didn't know, right, the notes I've given to you here are extracts from my entire set of curated notes, okay? Right, so it's all there one. I know exactly what's there. The cortex is this region here. So if you look at the diagram here, can we see this blood vessel connects all the way there, right? So this has to be the artery. Can we see that? Right, so a little bit more tidbits to share with everybody here. This is the cortex artery, brings it there. So after ultrafiltration, after selective reabsorption, it brings it back out like that. Okay? So a third way to identify our blood vessels. Okay? So if T is our artery, U is the vein. What's V? Ureter. Thank you. Okay. All right. So here we're looking at comparing T versus U only. T versus U only. Artery versus vein. Now, typically, you will hear the blood being described as filtered and unfiltered. Right? Which one has unfiltered blood? The artery law, because haven't go through the filtration process in the kidney. So unfiltered meaning dirtier. Well, because haven't removed the waste yet. So carbon dioxide, water, and glucose, which one is the waste? So here we're looking at carbon dioxide. But here if I'm not asking about urea at all, how? Why will these levels change at all? Why will these levels change at all? Respiration. Well done, Sonny. The kidney is made of living cells. So when blood goes into the kidney, treat the kidney as any other body cell. When I go to you, I have more oxygen. But when I leave you, I have less. Why? I gave you the oxygen, isn't it? So same thing for carbon dioxide. After I exit the organ, do I have more or less carbon dioxide? I should have more. So therefore, T should have less carbon dioxide. Going in, need to have less, come out then more, right? I collected more carbon dioxide, Then what's the relevance for glucose? If you think about function of the kidney, or it will be forced out during ultrafiltration, then what happens to glucose? We didn't highlight that just now. Or the glucose after ultrafiltration will be all selectively reabsorbed. Okay, then if I say all means like should be no change, right? But I don't have an option for no change there. So we have to think outside of this topic. Why will glucose levels change? Excellent. Respiration. The cells once more are living cells. They will use glucose for respiration. So now, T going in has more or less glucose than the one going out. More, I must have more. From here we got answer, isn't it? Then over here, that's how urine is produced, isn't it? So answer is B. Okay? Natalie, I'm trying to show that the artery has a narrow lumen. Okay, come. Cell G. It is this diagram that I believe Chloe was trying to explain to us. Alright now. What is cell G over here, Chloe? The phagocyte. We can see the cytoplasm pinching out, creating that heart of death, isn't it? Right? Engulfing it. So we use that to identify cell G as the phagocyte. Blood vessel E. What is blood vessel E? Blood vessel E is the capillary. Thank you, Annabelle. But what tells us that this is the capillary? One cell thick wall, you must continue with the wall, as well as thick is for wall. Wide lumen. And those 2 points that you've just given me is what B is asking. Okay? And the question says state. So you just have to state, yes. Polymorphous for cell G. Lymphocytes can also be polymorphous. So therefore it's too generic. Polymorphous for white blood cells. But now you have to identify lymphocyte or phagocyte. Then it doesn't apply already because it doesn't help me to differentiate. Okay? Thank you. Do you know what polymorphous is? Tell me. What does polymorphous mean? Okay, so if the polymorphous is not required in your syllabus, but if I'm encountering it, and since Fiona brings it up, polymorphous is we use simple English in our notes here, irregular shape. Okay, that's just it. Can change shape. That's what polymorphous refers to. Alright, so that's my suggestion is don't use that lah. Okay, Fiona? Awesome. All right. So what is happening at F and H? What's happening at F and H? So we choose F. What's happening at F? What's the cell that is within F there? The lymphocyte. So what do lymphocytes do? Produce antibodies. So you must bring in antibodies first, then you tell me what the antibodies do. Okay? Then for H, what is happening at H? Engulf and digest, breakdown. Thank you. Okay? Very straightforward question. Let's move on. For D, does a certain cascade come to mind today? Yes? Right? So think about that beautiful drawing that we did just now. From the start of the wound formation, the release of, what is that new enzyme that we learned? Thank you, thrombokinase. Thrombokinase, what does thrombokinase do? Prothrombin into thrombin. Do we remember the ion? Calcium ion. Thank you. So then what does thrombin do? Thrombin converts soluble first into insoluble. Okay? Then the threads, what do? They form a mesh and they trap red blood cells. At this point, trap red blood cells, what have we formed? A clot. What's the question asking of us? A scab. So then we go further. What are the two requirements for scab formation? Lose water and reacts with oxygen. Thank you. Okay? So we got very clear about the question. This one is scab. So go all the way to scab. If the question just says clot, do not talk about the scab. Yeah? Okay? We've all written this down already. So let's move on. Let's go. Question 12. Come, ENF. ENF. What's ENF? Actually, let me hear from you, please. Identify one.
The right atrium.
Wonderful. E is the right atrium. Lauren, F. Well done. Tricuspid valve. Okay, so E and F respectively, we have the right atrium and the tricuspid valve. Question is asking us, just look at stage 1 and stage 2. Look at the arrows. This arrow over here is from RA to RB. This arrow over here is from Clarine. For the blue arrow, what's this referring to? From what region to what region? Identify the chamber here. RB2. Pulmonary artery. And pulmonary artery. The question is asking us, describe the events which cause, why does blood flow in this direction? So if we just zoom into this, why does blood flow from RA to RV? Why is there a higher blood pressure in RA? Cardiac. Cardiac in the whole heart. So specify the muscles of the RA, isn't it? Right? So RA contract, then RV must do what? You must say, ma. If both sides contract, how? Both sides squeeze, cannot happen. RA contract, RV relax, blood flows through. Correct? But the question gave us a very important clue. What did they circle? What is F? So we must talk about tricuspid, right? What happens to the tricuspid? Open law. Can? Pause first, later I'll show you. Then here how? RV contract. Do we say anything about the pulmonary artery? Because it's just a vessel, no contracting or relaxing. Can? And then just now, what was the last thing we talked about? So here we also have to talk about something, isn't it? Semilunar. What's the name of this semilunar valve? Pulmonary valve. Semilunar valves are named after the vessels they are connected to. Right? So this is the pulmonary artery. This semilunar valve is therefore the pulmonary valve. Pulmonary valve opens. But then like that, 3 marks. We have written down 2 sentences. Because for the 2 arrows, right? Very good. Something that will be very easily missed out on. That is actually shown in the diagram. Is here. When the RV contracts, higher pressure, right? Higher than aorta. Correct direction. Okay, open. But RV higher than RA. Wrong direction. Your third marking point is tricuspid closes. Prevent backflow. Okay? This one, I will share with you. These are the 3 marking points. Draw then, Ray. No need for you. Okay. Which line shows us the pressure of chamber G? Can we identify chamber G first? Left ventricle. Next, from here, line 1 and 2, which one is left ventricle? 1, 2, or 3, sorry. Line 2, right? The ventricular pressure is always the one that increases and decreases the most drastically because what about the muscles there? It is the thicker, so it has the potential to be able to contract the most drastically and relax because it is the largest chamber, right? So it will do you good to also kind of be familiar with this graph. Atrium, ventricle, aorta. Because it is a ventricle that interacts with both sides, ma. Alright, not. The atrium is the one that only interacts with the ventricle. The aorta only interacts with the ventricle. So therefore, ventricle is the one that interacts with both sides. Okay? Alright? So it's just line 2, fairly straightforward. What is systolic? Contraction. Okay? Let me help you with this. Ventricular systole, the other word is diastole, right? So I typically teach this based on diastole. Why? Because diastole kind of sounds like dilate, and when we arrive at dilate, what results in a dilation of the vessel? Like relax lor. So whether atrial or ventricular diastole, it just tells me that that chamber is relaxing. Okay? Okay? Then you flip it around. So ventricular systole, oh, means the ventricles are contracting. When the chamber contracts, its pressure increases. Thank you, Lauren. So therefore, duration from the start and the end. When does contraction start and stop? If contraction results in pressure increasing, then you start with the increase lah. Why not? When does it stop increasing? Kind of here. Kind of here. Right? So pressure starts to decrease there already, isn't it? So what are we looking for? How much is that? Duration. Duration means you must math. 0.2, thank you. 0.3 minus 0.1. Right? Question asks for duration. Okay, so 0.2 seconds there. No need, Natalie, no need. Okay. All right, for question 13, easy. What are we looking for? Structure and explain to the function. What's the function here? What word tells me? Exchange, thank you. Once more, we get to exchange. We get to the topic of movements of substances. Right, so it's listed in the entire thing there, isn't it? The alveolar walls are very thin. The thin film of moisture is present. Dense network of capillaries maintains the steep concentration gradients. This is a very typical section B, the long question kind, right? Part A will be this 4 marks, then part B will be another 6 marks. Get this, easy 4 marks. Can? Describe and explain. Just need to bring in 2 features. Right? So it is also an important skill for us to have to figure out how much is the question asking of us. Yeah? Okay, breathing. Understand the graph over here? Question is asking us to simply look at A to B, which is here. So from A to B, look at the y-axis. Volume of air in the lungs increasing. So what stage of breathing is A to B? Is AB? Inhalation, isn't it? Inhalation. Question is asking, describe the changes taking place in the lungs from A to B. How to get 4 marks? Thank you. So therefore, this is all about the mechanisms of breathing. We talked about every exhale just now. You just flip it around. That's all. Correct? Right? You either do this, or if you learn the right thing, go ahead and do that. Both methods work. Can? Remember just now, the entire chunk of text that we wrote down, everything can be flipped except for the diaphragm, right? Cannot curve downwards, it must be relax, contracts and flattens. Thank you. And that's what the question is asking for. Can? Right? That's it. Sorry? Flip up. That one easy to flip up. You breathe in now? Inwards. What's the opposite of in? Out. What's the opposite of down? Okay. Okay, Sonny? Upwards and outwards. Sometimes you will see diagrams, they draw this. So we have outwards and upwards. Then opposite is for the exhalation one. Can? So the arrows like this. This is inhale or exhale. Inhalation, very good. And that's exactly what I want us to do. Instead of memorizing stuff, Just breathe lah. You just feel yourself. Oh, this is inhale. This is exhale. That's all. Thank you, Chloe. All right, come. CD now. Question is asking us to focus on CD. So, what is this person doing? Question is asking, describe the change first. Then we suggest why. So, what are the changes we observe from C to D? From C to D, what are the changes we observe? We need to describe two different things. Two different things. Shalice, can I have a view, please? C to D. There are two changes that occur. Can you tell me one, please? Can. Increase in the volume of air in the lungs, meaning if you just bring it together, depth. You're breathing deeper, isn't it? Okay, so that's one change. Breathing deeper. What's the second change? Faster. That's rate. Can. So breathing deeper, breathing faster. Yes, I will, Kate. Right? Breathing deeper, breathing faster. Why? Question just says, suggest why. Why will your breathing become deeper and faster? Exercise not enough, must say vigorous exercise. Can? Because jogging also exercise ma. But if jogging you like that, then okay, so must vigorous exercise. Awesome, thank you. Who's the fitter athlete here? P and Q, who is the fitter athlete here? Sorry? YP. Requires less? No, this is a monitoring. Athlete P is the fitter one. Why? Sorry? So how do we know from here? Very good. Athlete P's aerobic capacity longer. So athlete P could undergo aerobic for longer, then anaerobic kicked in. Fair? Can we see that? Right? P is the fitter one, and then this is due to training and stuff like that. Okay? This one is just graph skills, so I'm not gonna talk about that. And then the suggest the difference is what Sonny mentioned also, is just training, larger lung capacity, maybe something like that. Can? Okay? Larger lung capacity means you take in more oxygen, breathe in, absorb more oxygen as well, so that works. Okay? Alright, so these two questions fairly straightforward. I'm not gonna spend too much time on it. This okay. If you're wondering, right, these kind of things are when you are exposed to new terms, they will explain to you one. So don't panic. It's just there. Okay? So it's when it increases sharply. Then just increase sharply is here ma, for each of them. Clear? That's all. Right? So only at 10 kilometers per hour, Q's threshold was met. But P must hit 12, then the threshold is met. Okay? So just read graph only. What do you think part 3 is asking of us? How do we have 4 marks? Describe and explain the changes in P. Look at your graph. Just look at P. How do you describe the change? How do you describe the change? This is athlete P's curve. How do I describe it? Is there a way for me to break it down? How many parts are there? 2. Very good, right? I have here, I have here. Okay? But whenever a question is accompanied by a graph, what else do we need in our answer? Values, data. So describe the curve, you must give me trend and data. Increases gradually, data. Increases, this is what happens when you talk for 5 hours. Increases sharply, data. Can? Then why? Then you talk about what's the process then? What comes up? What's the process here? Anaerobic respiration. Can? Right? So if I just clue you in over here, what are we thinking of? We have increases gently, then add data. Increases sharply, add data. Then last but not least, we explain anaerobic respiration. And this is worth too long. Can?
Why did you have to explain aerobic as well?
What is the y-axis?
Lactic.
Lactic, right? So when we see, we focus into lactic acid. If this was a 6 mark, then maybe I will talk about aerobic a little bit as well. It's always about choosing what is the priority in the question there, right? Wonderful question, actually. Thank you. Okay. J and K, what are they? Cilia cells. So cilia is this, but the cell that has cilia, I gave us the name there, isn't it? We can go for a ciliated cell, or the full name there, ciliated epithelial cell. But the word has to be ciliated. It must have a cell with cilia. Ciliated cell is the least that we can accept already. Cannot say cilia cell, Thank you. So J is the ciliated cell. What's K then? K is the easiest name. K is not mucus, K is a cell. So therefore, K is a mucus producing cell. Okay. So when the question asks us to state how they protect the respiratory system, whose cell's function should we state first? The mucus one, right? Remember, mucus tract first, then you say the cilia sweep. You could talk about the cilia sweep, but then you must kind of like backtrack. So mucus first, then the cilia sweep. Okay? Awesome, that's it. Smoker, decrease in the volume of air per breath. So this one we are focusing on harmful effects of cigarette smoke already. I know we're out of time, so let me just very quickly finish up this one. What is the which component of smoke are we talking about here? Tar, thank you. Right? Tar blocks the airway. Right? So I mentioned this just now. The smokers have a higher resting breathing rate, isn't it? Right? So this one, you can just quickly pen this down over here. Okay, remind yourself, tar. Okay, this one, what do you write down? Well done, carbon monoxide. Okay, carbon monoxide. Question 15 is actually very table skills only. Definition, nothing too fancy. I asked us about D just now. I asked everybody about D just now. Why no proteins in urine? Protein. Too large, thank you. Correct. Glucose, then is the one that is all selectively reabsorbed. Can? Okay, everything is there already. So that's why I spent a little bit more time just now and less time here because the answers are all there. Okay? Last part over here. We focus on the word diuretic. But in our syllabus, what is the thing we learn? Anti-diuretic, isn't it? But it's simple, right? Opposite law. What does that anti-diuretic do? Increases permeability. Diuretic decreases. Can? Flip it around. The moment you can get here, everything flows.
Say increase urine production.
Absolutely. But how? Then you talk about the permeability and absorption one. And that's actually what, well done, Sonny, Part B is asking of us. Okay? Why Rain's urine production increase? Rain is the one that drank the green tea. This question is telling us green tea contains diuretics. Okay? Can we identify the two boxes there? Afferent, or how? Which one is the A one? Left side or right side? Right side. The wider lumen is the afferent. The narrower lumen is the efferent. Can? Question is already labeling for us. Glomerulus here. Bowman's capsule here. So what's the process? Identify the process first. What process here? Ultrafiltration. Describe it. Then we talk about the difference in pressure. What word do we use? Difference in lumen generates pressure. Force. Thank you. I heard that word there. Forcing what out? We wrote that down just now. Forcing what out of the blood? Small solute, water and small solutes. Thank you. Small molecules work as well. Can? All right.
Can we say blood plasma?
Cannot, because there are things in plasma they are not found here also. Okay. So this one is then the difference in lumen diameter. Then we talk about the pressure generation. Can the question specifically asks, hey, the C just now you identify one. They talk about that. Okay. Okay. Yes. All right, so that's the last one already, right? No more. If you flip it, you see the one. That one is for the combined bio people. So that's it. That's all we have for today. It will greatly help if you all can do me a favor over here. Just get your phones out and just scan the Tampines side because that's where we are at here. And just show me that you have done it. I'll pass you all the beautiful foolscap pad over here. For those at home, I would really appreciate it if you can do the review for me as well. And hey, if next time you pass by any of our centers, show the review, you can get the foolscap pad as well. Okay. As you are doing that review, please make sure to think about what we have talked about today. The answers will be provided afterwards. So try, especially for the last few questions. We just jot down the points at the side only. Go and try them first. Try and word out your answer. Then refer to the answer provided. Okay? It'll be available online. Okay? Great. Here you go. Have you all scanned it? Just waiting for Sony as well. Welcome.
But it seems like too many redirect.
Then refresh again. Awesome. Here you go. Welcome. Yeah, do it up. Scan it up. Okay, so just let me know for this part over here. The registration form for joining in my class next year is already open already, right? I need you all to understand that this is just an interest generation thing. Not required to start right now. Nobody's gonna start right now. Okay. The idea that I need you to understand here first is typically our academic year starts at mid-November. Right, but I understand that people are going on holiday and stuff like that, so that's absolutely fine. The thing that you're looking at here is all the early bird perks. But even without the early bird perks, you register, you can secure your slot in my class first. Okay? I have classes here as well as in Kovan, so take your pick. Alright, take your pick. Make sure that the earlier you start with me, after your holidays and stuff, go for your well-deserved holiday. The earlier you start with me, the sooner we can plug in those gaps. And from what we have done together today, we have quite a few gaps that we can work on together, right? So after today, go and have a good think about it. After today, think about your end of years, and I hope to see all of you next year working towards our O levels as well. Okay? Right? If not, thanks for learning here with me. I hope I've helped you fill in some of the gaps, understand things a little bit better. Show me your reviews and I'll pass you a foolscap pad. And for those at home, thanks for learning with me here. I hope that you will be able to follow along well as well. But if not, that's all. Goodbye, everybody. Come.
Third.
Why trick? Reject your request. No, and then let's go. Let's use it.
Can I have that?
Here you go. Here you go. Very strong. Anymore? So ask me, can. They just thanks. Either this direction or
Or like
either
do another
wrong
one.
That's true. How do you know? We can't see. Okay, yang belum kawai okay. It's the same material eh. No, I only bring one kawai.
Yeah.
Okay.
Maybe something very structured. Yeah, that's good.
Yes. Annabelle, is this? No, Annabelle. Next is. Alright, those at home, right? If you are not nothing else, you can go. Thank you. Kate. Yes.
Mr. Fang,
Yes, hi. Who's
can you
that?
send the answer sheet, please?
It'll be available online, not right now. Hi, yes. Okay.
Okay, thank you.
Great. Welcome. Okay, so question 4 again. Starting, when a diaphragm starts to contract, the pressure will start to decrease. But if you choose point A, the pressure is very low already. It cannot decrease any further. 10K? But it's in line now with the timing. The timing is all in line. It's in line with the timing one. At point A, the pressure is very low already. But diaphragm starting to contract means the pressure must start to decrease also. And that's not what is happening. So everything must align. Because it's based on the x-axis, it's aligned with the time also. So if we choose point A, which is here, pressure is already very low already. So this point is actually contracted fully. This one is contracted fully. A is when the diaphragm has contracted fully already. That's why immediately volume goes in for inhalation to occur. So if you look at this graph, is one cycle per breath, one complete breath. This is another one. So the next one is what? At this one, decrease, then slowly increase again. So it's a cyclic thing. Okay? Ask me, yes?
Inhale, pressure, increase.
So that air can come in.
Then shouldn't the volume increase? But at C.
So it's later. That's why it's starting to. The word is starting to. Inhalation hasn't occurred yet. Can we think about it as four parts? The change, the inhalation, the change, the exhalation. So the starting to is important. Yeah. So I put this question because this one tends to be the one that tricks people a lot. So the starting to is important. We say inhale, exhale as one complete cycle, but then the muscles and the diaphragm, everything, then the inhalation, then the action, then the exhalation. Okay, then this one goes further than your syllabus, right? That changes occur before air moves in or moves out. So it's like a change, change, like that. No, my class says 6.
Can you show the part that got like the scat one in the notes because I'm not copy that part.
Yeah, that's
At the very start.
it. Give me a
Thank you.
second. Actually, all the recording is going to be available afterwards. So
Okay.
yeah,
Thank you so much.
where is my scat? Oh, here. Then come, ask me. Welcome. Rochelle as well, yes, and then Joel. Okay, yes, hi.
How do you know which the inequality sign to go where?
Woo. Wrong. Where's my graph? Is that right? Is that right? No. It's the notes part or the question part. Question. This one ah? No, or MCQ, MCQ.
Same-
Yeah, it's the same thing. Time. So aorta pressure increases below left ventricular pressure. So it's about the change, not the point. If you look at the point, then same lah. That's what intersection means, right? But it's always about the change. So intersections, then you look at the change that occurs at that intersection. If you look at them in terms of math, they're equal and cannot answer it. So you must look at it in terms of the change.
Wait, one more question. Wait, for this one, P is,
Mucus producing cell.
but isn't this the mucus?
But they're telling you what? They tell you it's cells already, ma.
I thought this was the cell.
These are the ciliated epithelial cells. This is the mucus producing cell that will spill mucus out. Should I have chosen a nicer diagram? No lah, I think it's fine. Yeah, yeah.
Okay. Also, what's the answer key?
Yeah, it'll be available online afterwards. Let me check out how I'm supposed to distribute the answer key. It'll be a soft copy thing. Okay, so you let me just check in how it will be distributed to you all. Maybe it will just be like thrown online. Then you just grab it on your own. Okay.
From the last lesson, they will show a QR code at the end. We will scan.
Yeah.